Chapter 13. Photosynthesis in higher plants. Summary. Green plants make their own food by photosynthesis. During this process, carbon dioxide from the atmosphere is taken in by leaves through stomata and used for making carbohydrates, principally glucose and starch. Photosynthesis takes place only in the green parts of the plants, mainly the leaves. Within the leaves, themsophil cells have a large number of chloroplasts that are responsible for CO2 fixation. Within the chloroplasts, the membranes are sites for the light reaction, while the chemosynthetic pathway occurs in the stromatal aperture. Photosynthesis has two stages, the light reaction and the carbon-fixing reactions. In the light reaction, the light energy is absorbed by the pigments present in the antenna, and funneled to special chlorophyll molecules called reaction-centric chlorophylls. There are two photosystems, PSI and PS2. PSI has a 700 nm absorbing chlorophyll AP700 molecule at its reaction center, while PS2 has AP680 reaction center that absorbs red light at 680 nanometers. After absorbing light, electrons are excited and transferred through PS2 and PSI and finally to NAD-forming node. During this process, a proton gradient is created across the membrane of the thylakoid. The breakdown of the proton's gradient due to movement through the F0 part of the ATPase enzyme releases enough energy for synthesis of ATP. Splitting of water molecules is associated with PS2, resulting in the release of O2, protons, and transfer of electrons to PS2. In the carbon fixation cycle, CO2 is added by the enzyme, rubisco, to a 5-carbon compound RUP that is converted to 2 molecules of 3-carbon PGA. This is then converted to sugar by the Calvin cycle, and the RUP is regenerated. During this process, ATP and NADPH synthesized in the light reaction are utilized. Rubisco also catalyses a wasteful oxygenation reaction in plants, photorespiration. Some tropical plants show a special type of photosynthesis called pathway. In these plants, the first product of CO2 fixation that takes place in the mesophyll is a 4-carbon compound. In the bundle sheath cells, the Calvin pathway is carried out for the synthesis of carbohydrates.